Hello, in this video, we're going to talk about neuromuscular junction. Okay, so the neuromuscular junction is a chemical synapse between a motor neuron and a muscle fiber. We have them obviously everywhere throughout the body because we have many neuromuscular junctions in all of our whole muscles all throughout the body. So we have many of these chemical synapses. So an action potential in the neuron, and we'll talk about that in a future lecture. We'll review action potentials and nervous system function. But an action potential in the neuron causes release of acetylcholine. That's a neurotransmitter that we use here and in many other chemical synapses throughout the body. So we release acetylcholine from the synaptic end bulb. So those are the little end projections of the axon terminals, which are branches of the axons of the neuron. So if we look all the way at the top picture there, you see there's a branch coming down from, or sorry, not a branch, there's an axon coming down from a motor neuron, and then that divides into the axon terminals. Those are the little branches, and at each end of the axon terminals, we have a synaptic end bulb, like the little feet at the bottom. And those little feet, those little synaptic end bulbs, are what we are zoomed in on in these bottom two pictures. So we're seeing the synaptic end bulb where it interacts with the muscle fiber that neuron is controlling. So we have a neuromuscular junction at each of these synaptic end bulbs. So there are multiple per every motor neuron at every muscle fiber. So you can imagine how many millions we must have of these in the body. Acetylcholine travels across the synaptic cleft, which is the space between the synaptic end bulb and the motor end plate. So it travels across the synaptic cleft to the acetylcholine receptors located on the motor end plate of the sarcolemma of the muscle fiber. Okay, so remember the sarcolemma, that's the cell membrane of the muscle. The motor end plate is the area of the sarcolemma that contains the acetylcholine receptors. Okay, so in this middle picture, the motor end plate, it's that sort of white line that's lining the sort of wiggly shape there. So that white line is the motor end plate, and it contains the acetylcholine receptors, which are those sort of little coffee bean looking structures there. They're sort of purple coffee beans in this picture. So those are the acetylcholine receptors. So the synaptic end bulb has these vesicles. That's what those little bubbles are inside of the synaptic end bulb, that they're like little bubbles full of the acetylcholine. And so when an action potential travels through the axon of the neuron, it signals release of those vesicles containing the acetylcholine. So those little bubbles, those little vesicles, get pushed out of that synaptic end bulb, and the acetylcholine is released into the space in between, the synaptic cleft. And that acetylcholine travels across and binds with the acetylcholine receptors, and then it triggers an action potential in the muscle, just like we just had in the neuron, by causing ion channels to open in the sarcolemma. Okay, so the acetylcholine causes opening of the ion channel. So until the ion channels open, the cell is polarized, meaning that it's very negative inside the cell and very positive outside, or much more positive outside the cell. So it's polarized, meaning like we've separated the two charges. And so that means that it has a great potential to carry that sort of electric current. So when we open those ion channels, it allows the sodium ions, which have a positive charge, it allows those sodium ions to flood into the cell, and then that's what is initiating the action potential. And that's the same in both the neuron and in the muscle cell. So this effect at the neuromuscular junction is very short-lived, because the acetylcholine is released into the neuromuscular junction, but very quickly, if we don't have another action potential to continue to release acetylcholine, very quickly that acetylcholine is taken out of the neuromuscular junction so that those ion channels don't stay open, they close again. And so there are a few different ways that we get rid of that acetylcholine. One is that it's broken down by an enzyme, acetylcholinesterase, And keep in mind, anything that ends in ACE, ASE, is an enzyme. So acetylcholinesterase, that is an enzyme that specifically breaks down acetylcholine. It also, the acetylcholine could be taken back up into the motor neuron. So it could actually be taken back up into that synaptic end bulb, and it'll be recycled. It'll be reused by that cell um, for the next signal to contract. It also could be taken up by neighboring glial cells or neuroglia, and we'll talk about those later on when we talk about the nervous system. And then the third option is that it could just simply diffuse away from the synaptic cleft. It could simply just diffuse away into the extracellular fluid within the tissue and just drift away and no longer cause any action potentials in the muscle. All right, so that's all I have for you. Thanks for watching.